Okay, good morning everyone and welcome back. Today we're going to talk about part 1b of sugars and nucleic acids. This is going to be about the prebiotic synthesis of nitrogenous bases. So this is the second portion from our outline. So yep, as I said, prebiotic synthesis of nitrogenous bases. So like we did with the peptide unit, we described some evidence where we could take these basic molecules that were around in the universe, and if you mix them together under very harsh conditions, you could actually generate some amino acids. So that was quite interesting. What about things like nitrogenous bases, right? Those are ubiquitous. Can we actually make those as well? So this is taken right from the textbook, but the question is, can we take some of those basic building molecules like formaldehyde? Recall, formaldehyde was something that was generated during that Urey-Miller experiment in the synthesis of glycine. And hydrogen cyanide, another basic building block. So could, these carbon units be used to make ribose, which we know is the basic sugar backbone of DNA? And could the hydrogen cyanide units be used to make heterocycles, things like adenine? And then later on, there'd be a way to join them together through that oxocarbenium ion that we talked about last lecture to generate the N-glycosidic bond adenosine, right? So if we think of, what was around prebiotically, we had things like water, hydrogen cyanide, ammonia, and then we also had things like methane. Okay, so the synthesis of bases, there are actually many different methods in heterocyclic chemistry. It's a fairly rich field in organic chemistry, and a lot of different complex heterocycles can be made from different small building blocks. It's been shown that maybe if you take things like ammonia, hydrogen cyanide, that you can get polymerization of hydrogen cyanide, and this can be the precursor to make adenine, right? But there's so many different ways and combinations to make this, and it's still really not understood why adenine and guanine and thymine and cytidine and the classic bases that we know are the ones that actually survived and are the ones that we use. Okay. So I'm going to go through some evidence showing that the synthesis to generate purines like adenine can be done using a simple molecule called hydrogen cyanide. Okay, so synthesis of precursor to generate purines. All right, so let's take a look. All right, so let's take a look at hydrogen cyanide. So here's hydrogen cyanide, and here's another molecule of hydrogen cyanide. What we're going to do is we're going to get these molecules to come and react with one another. Hydrogen cyanide actually is a very good acid, so it can donate protons. It's also actually a base as well. So let's just do sort of a classic acid-base chemical reaction, and then what you're going to end up with is the nitrile anion plus this hydrogen cyanide that now has a nitro group that is protonated and is now actually a good electrophile. So if you look at these two molecules here, you now have a good electrophile at this carbon, and you actually have a good nucleophile at that carbon. So, so hydrogen cyanide is actually really good at sort of self-reacting with different molecules of itself based on simple nucleophile electrophile coming together. So once that happens, then we generate this molecule here that has this imine-like functionality. So we've literally just had these two molecules come together, and at this point we could ask ourselves and do the same thing over again. So this nitrogen here is basic, and it can pick up a proton from hydrogen, a different molecule of hydrogen cyanide, and just do another acid-base reaction. And what you've done here is you've created like an iminium ion, which is very electrophilic, and at the same time generated a cyanide anion. So like we'd seen in the second step, the nucleophile here will react at the electrophilic portion, get electrons moving up. All right, so let's see, what does that look like? Okay, so we have this molecule here that has now sort of one carbon, 2 carbon, 3 carbon, species 1, 2, 3, and you know, so we're kind of building 1 carbon at a time. So we're going to do the same thing again, pick up another proton from hydrogen cyanide. This will come down, and you might be saying, well, where is this guy going with the synthesis, right? Referring to me, like, what am I doing, right? So, you know, so this will come and back and react. And in a second, I'll show you what our target molecule looks like. And our target molecule looks like something based on computational models is one of the precursors to make adenine. So a little bit more on that in a second. So just bear with me. So this comes and reacts, right? Just like we've seen before. So you can see this pattern. And what we have what you have is a molecule that looks like this. Now the molecule that we want to end up making, okay, it's not adenine yet. Like I said, it's a precursor to adenine, is this molecule here. Okay, it's called diamino malonitrile. Okay, so that's a little bit of a mouthful, okay, but that's what we need to make, and we're kind of almost there actually. So if you look here, and I've drawn in this proton just to sort of help with what's happening, is that now you've seen cyanide anion always acting as a nucleophile, but you can now imagine this group coming here and via elimination actually forming that double bond that's needed for diamino malonitrile, and therefore picking up a proton on the way out from hydrogen cyanide. Okay, so that will give you this target molecule here. Now, if you say diamino malonitrile, whoever you know, found this molecule based on computational methods, as I'll talk to you in just a little bit, so that's just too hard a word to say. Okay, it's actually abbreviated as DAMN. So whoever made that acronym must have had a lot of fun or was in a really bad mood. Who knows? Anyways, so that's the DAM molecule that needs to be made before we make adenine. But really the take-home message from this slide is that you can see that you can make these complex molecules just using hydrogen cyanide. So hydrogen cyanide acts as a base, right? Also acts as an acid, nucleophile, and electrophile. So it really does all 4 things that, you know, most molecules can only do one thing. So base, acid, nucleophile, and electrophile. Okay, so that's the mechanism there. Want to walk you through that because if you kind of just look at that really quickly, it can seem a little overwhelming. So what I have on the next slide here actually is right from your textbook and actually describes exactly what I just talked about. Okay, And I really believe in active writing and going through the mechanism as I teach it and explain it, rather than me saying, okay, well, here's the mechanism. Okay, but you can see it's comprised of the acid-base step, so that step was actually skipped at the beginning. Here you're just showing the cyanide actually attacking, so I actually prefer the way I did it. Okay, and then, but as you can see, they get to that same target molecule called DAM. All right, so why are we talking about this molecule so much, this DAM molecule? So it's been shown that this molecule here that we just made can be actually made at 25 degrees in water, so fairly mild conditions. And in the presence of UV light, which you can imagine was around, because of the stars, well, still is around, this can form this compound here called AICN, which stands for amino imidazole carbonitrile. Okay, so another mouthful. That's what this molecule looks like. And then later, hydrogen cyanide can be used to form things like adenine from this molecule here. And if you take this sort of urea type molecule here, can actually make things like guanine. So again, other simple building blocks can be used to make the purines that we use and know about today. So let's see how that mechanism works. But it's really not super clear how this transition forms, okay? There's been some computational evidence showing that it goes through some very sophisticated rearrangements, perhaps. Okay, but more on that in a little bit. But let's take a look at this step up here, right? How do we actually make adenine from this molecule here? So let's look at AICN. Okay, and again, we're using hydrogen cyanide. And let's see, what we're going to do is we're going to, let me just get my bearings right here. Yeah, so this group here is actually going to come and it needs to add, the carbon needs to add to that. So as you already learned previously on last slide or 2 slides back, you can just do that acid-base chemistry, works really nicely. And you generate a really nice electrophile in the meantime. So this nitrogen can come here and can react to form that key nitrogen carbon bond, which is needed here. Okay, so once that happens, what we're going to do is that's going to come and react okay, and you're going to end up with something that looks like this. And then you're going to get a proton transfer, right? You're going to lose this proton. So maybe we'll get cyanide to come here and remove that proton, because it can also act as a very good base. And so we have this. All right. Now what we need to do is we need to find a way to close this ring to make it a 6-membered ring. So if we count from this carbon, you have 1, 2, 3, 4, 5, 6. Okay. This nitrile group, as you know, is a good electrophilic partner. This nitrogen can be a good nucleophile. But to make this a better electrophile, what we're going to do is we need to protonate that, right? So where do we get a proton from? Our good friend hydrogen cyanide. Well, I say good friend with respect to the synthesis, but as you should know, hydrogen cyanide is actually extremely toxic. So don't ever come near it if you don't have to. It will poison your cytochromes and cause respiratory failure. So pretty nasty stuff, okay? But it's definitely your friend when it comes to this mechanism. So now the nitrogen comes, reacts here intramolecularly, And we now have our 6-membered ring. Nitrogen's protonated there. And so what we need to do is we need to simply move a double bond here, because if you look at adenine, there's a double bond here. Got to move this pi bond here, and then this has to become NH2. So at this stage, it's a lot of proton transfer movements. And again, I'm always following the organic chemistry rules. So I'm going to pick up that proton, form a double bond here. If I do that, I have to break that pi bond. Okay, so now we're going to form a double bond there. And then this will come and pick up, a proton from something like hydrogen cyanide. And that will give you adenine. Okay, so experimental evidence, so you can actually do this in the laboratory. Again, don't do this because it's hydrogen cyanide, but you can generate this molecule here, UV can convert it, and then you can get, even though it's a low yield, you can actually generate adenine guanine. So it's very likely that these were formed prebiotically, perhaps using a very similar type of methodology. Now, what do we know about the photochemical steps involved in the prebiotic synthesis of purine precursors from hydrogen cyanide? So there's been a lot of interest in trying to understand exactly what's happening, and a lot of things have been done computationally. This is a paper that was published in Angewandte Chemie. It's a leading journal in chemistry, and essentially the authors of the study investigated the mechanism of photoconversion of the hydrogen cyanide tetramer into a precursor of purine nucleobases and nucleotides by computational methods. So this was all done computationally. No wet lab stuff was done. But this was generated a lot of excitement because this is one of the first papers to show that you could have these key precursors like DAM, that was showing that this was an important precursor in the synthesis of those purines. Okay, this is a fun sort of mechanistic slide from that paper showing that when you start with DAM, your DAM molecule, and you need to get to AICN, okay, it's very complicated in terms of how that happens, and people really didn't understand what happened with respect to when UV light, when this molecule DAM was incubated with UV light, how it actually converted to AICN. This computational work highlights that there's definitely an energetic pathway, a downhill energetic pathway leading to that structure, and it could occur through several different derivatives. So you get rotation, flipping from like cis to trans, then you can get this lower energy molecule here, and then you can have this like strained 4-membered ring, which kind of helps to split that open to generate the molecule here. Now, would I ever expect you to be able to recopy this? Probably not, okay? Very, very slim chance. I just want to show you that there's a lot of interesting computational work done in this field to try to understand and capture what happened many, many years ago. All right, so we looked at A and G. Other polymers based on hydrogen cyanide include things like cyanoacetylene, cyanogen, and you can generate some of the other molecules like some of the pyrimidines. And there's also some other sort of rare heterocycles as well that are used in nucleic acids. So, on some Friday evening, please try out these mechanisms on your own. You might actually have some fun. Okay, some other things I want to talk about, going back to pyridine, properties of pyridine. Okay, so last lecture we talked about it acting as an acid and a hydrogen bond acceptor. Pyridine can also actually act as a nucleophile and can react, with these acyl halides Electrons can come up, and then you can lose your leaving group to generate these intermediates here. And pyridine can also undergo these types of substitution type reactions. And what's nice is that you have a good leaving group attached to these, but it's still fairly stable because it is aromatic. So properties of pyridine and pyrimidine. Okay, so I want us to think a little bit about a balance between aromaticity and charged forms versus non-aromatic and neutral. And we're going to talk about a molecule called NADH, which is a molecule that you've heard of before. I'm going to draw it out. It's nature's, it's nature's reducing agent. Okay, what does it actually look like? So you've seen like NADH, NAD+, so many times in metabolism and biochemistry, right? But again, we're chemists here. We have we're going to zoom in on the chemical bonds here and try to understand the structure of what it looks like and why it actually works that way. Okay, so this is NADH. You know it's a good reducing agent. That means it can transfer a hydride atom to something that can be reduced. So I'm going to draw a functional group ketone. Ketones are very prone to reduction. And if you think of how this is going to happen, how are we going to transfer a hydride to here? Well, the hydride is going to come from up here. Okay, but if the hydride comes and attacks, that doesn't seem very favorable because you're going to be left with a positive carbocation there. That's not so good, right? Now, however, you have a compound here, right? Is this an aromatic compound? Well, you have 1, 2, 3, 4, 5, 6 electrons. So you say, oh, I got 6 p electrons potentially, but you need to have that in a circular network. So this molecule here is not aromatic. Okay, if we want to get reduction, we're going to need some help from the nitrogen here. So electrons will come, form a double bond. That has to move up to here to form another double bond. And then you get this hydride now in a position to attack that ketone. Okay, so what that will do, of course, is that will, with a proton transfer, transform that ketone to an alcohol. But what happens to the NADH? What happens to our reducing agent? So let me draw what happens. Let's just follow exactly what we did with our arrows. So we have a double bond here. We now have a double bond here. Now we have this compound here, 4 bonds of nitrogen positive. Is this an aromatic compound? Yes, it is. It looks just like pyridine, which we've been studying for a while. So the driving force actually to get a reducing agent from a heterocycle that's attached to a sugar like this is that you're going from a compound that's not aromatic, but is a good reducing agent, to something that's aromatic. So that's the driving force for this reaction. And then similarly, you've learned that this is reversible. So you can imagine this is now easily oxidized. So you can actually, this is the oxidant, and you can go back to your reduced form. Okay? So it's a balance between aromaticity and charged forms, right? So it's quite interesting. That's what nature uses in metabolism to generate, nature's way. You can think of this like, sodium borohydride or lithium aluminum hydride, but a much gentler form, something that's biocompatible. Okay, that's the chemistry behind it. And lastly, the last slide for today is just giving you some interesting perspective on this, this NADH, this nicotinamide moiety here, that's really responsible for redox chemistry, as we just talked about. But is it just, some random molecule that formed? Well, it's been actually shown that you can take these basic building blocks and generate these two basic precursors, okay? Compound with a single double bond, and then this nitrogen-contained compound with two double bonds. And you've learned about a reaction, a cycloaddition 4+2 reaction in organic chemistry called Desaulniers reaction. What that is that you're taking a diene, so two molecules with a dienophile, and essentially through a cyclic conversion process, you can actually get a 6-member ring forming. And so actually, if you put these two together, electrical discharge may have created the energy needed to generate both these molecules, but then also the Desaulniers, and you can generate this molecule here. And then it's fairly straightforward to oxidize this nitro group to this moiety here. And although people have done this and they had a 1% yield, if you have time on your hands like the universe has had, then you just don't need a lot of it. And they're basically nature's redox electrochemical switch. And this could have been around long before enzymes were involved, right? So there's been a lot of debate in the field in terms of, well, what was made first, right? Was it proteins and peptides that could catalyze reactions, or was it kind of nucleotide precursors like this, simple molecules that may have been around to conduct redox chemistry? Who really knows, right? But certainly you can see that a lot of these molecules are not made by coincidence, but are made from the basic building blocks that were around in the universe. Okay, so that's our unit for today on prebiotic synthesis of nucleosides and derivatives. All right, have a great day.